Excited to present to you a little bit about our work on multi-omics analysis in biliary cancers to help define novel molecular subtypes in these cancers. So Dr. Goyle gave a really wonderful overview last night of cholangiocarcinoma, and I think the really important takeaway message is that these biliary cancers, which encompass intra and extrahepatic cholangiocarcinoma, gallbladder cancer, and ampullary cancers, really demonstrate significant molecular heterogeneity across and within these anatomic subtypes. And at this point, we're treating these tumors the same in the first-line advanced setting, but while the second-line target therapy space is rapidly evolving, there's still room to improve and have more refined strategies. So this really highlights the need for developing a functionally relevant classification system that extends beyond anatomic subtypes and genotypes. So there are very limited models in biliary cancers. So one of our primary goals was to establish these models, characterize them deeply, and be able to use them to study this complex biology in depth. So we compiled a collection of 63 cell lines, which included 30 novel patient-derived models, the rest of which were sourced from across various institutes. And then we performed multi-omics analysis, including DNA, RNA, proteomics, and whole genome CRISPR screening. And this biliary cell line atlas really reflects the spectrum of genomic diversity and anatomic subtypes that we see in these tumors. And for example, in line with what we see in patient data, we see that FGFR2 fusions and IDH1 mutations are primarily observed in intrahepatic cholangiocarcinoma. And on the other hand, in extrahepatic cholangiocarcinoma and ampullary models, we see more KRAS mutations. So one of the key questions was, how well do these cell line models reflect the biology and molecular circuitry of the patient tumors from which they were derived? So we looked at the transcriptomes of 20 cell lines and their associated patient-derived xenografts. And here you can see that the cell lines, which are the circles and, the triangles are the cell lines and the circles are the PDX tumors, they really cluster closely together as indicated by these pairs and dotted lines. And in this cohort, 85% of the cell lines had their nearest neighbor as their corresponding PDX. And that really indicates that these cell lines are quite representative of their patient-derived models. So next, we compared the transcriptomic data from our biliary cell lines with 200 plus cell lines derived from other GI cancers. And on the left, in the orange, you can see that these biliary cancers really displayed a very distinct clustering pattern, reflecting their unique transcriptomic signatures. And they cluster somewhere between HCC and pancreas, which reflects their shared developmental origins. And then on the right, we extended this analysis to patient data and found that these biliary models cluster really tightly and recapitulate the transcriptomic programs of the primary patient tumors. As an approach to identify novel and functionally significant relationships, we clustered these cell lines based on their transcriptomic profiles. And here we actually included HCC based on their similar origin. And what we found is that there's 4 distinct RNA subgroups labeled R1 through R4. And here labeled on the heat map, there are 2 clusters of biliary cancers, R3 and R4, which have unique signatures. And somewhat interestingly, this R3 signature seems to have overlap between R2, which is primarily HCC, and R4, which is exclusively biliary cancers, suggesting that there's some sort of intermediate phenotype here. So we know that there's this concept that hepatobiliary cancers encompass a spectrum between hepatocyte-like and bile duct-like phenotypes. And on the left, as a way to examine the relationship between cell lineage identity and our RNA clusters, we ranked the cell lines based on the expression of both bile duct and hepatocyte markers, and found that biliary cancers harbor different cell lineage programs, which we designated EMT, hepatocyte-like, bilineage or bipotential, and ductal-like cancers. And on the right, you can see that these novel classification systems really extend beyond genotypes and include some of these common markers. In the bipotential group, we see IDH1 and FGFR2 alterations, whereas in the ductal group, which has more of a mixed phenotype, we see KRAS, BRAF, and HER2 alterations. So we tested these lineage classifications in a patient cohort looking at overall survival in groups that were enriched with the R3 bilineage and R4 ductal signatures. And as you can see, looking at the Kaplan-Meier curves here, patients that had higher R3 signatures had improved overall survival, whereas those with the R4 or ductal signature did not. And finally, we also grouped these cell lines based on their dependency profiles using whole genome CRISPR screening. And I wanted to highlight one really interesting subgroup that we identified, which was defined by this really strong dependency on the driver of squamous differentiation, TP63. 2 out of 5 of these models were derived from adenosquamous carcinomas. And up here on the top right, you can see that when we look at the transcriptomic data from patient tumors, there's this correlation between TP63 expression and this TP63 network that we see in this specific cluster. And we found that this specific type of tumor was seen in about 5 to 10% of biliary cancers, which was higher than expected. Not surprisingly, the survival with P63 in this cohort was worse. And so while these adenosquamous carcinoma variants are quite rare, it's possible that there is sort of more partial squamous differentiation biliary cancers. So in summary, we've identified functional subtypes of biliary cancers based on transcriptomic, genomic, and dependency profiles. And the hope is that this will help improve the treatment landscape in biliary cancers. So thank you very much to the Bardisi Lab, and particularly want to recognize Dr. Vindhya Vijay, who spearheaded all of this work, as well as the Gadi Getz Lab in the Broad. Thank you.